On the 2nd of April, 1953, two men walked into the Eagle pub in Cambridge, England, and one of them announced to everyone present that they had found the secret of life. Francis Crick, James Watson, the double helix, the structure of DNA. The announcement, informal, slightly drunk, entirely characteristic of the man who made it, was followed by a paper in the journal Nature on the 25th of April, 1953. The paper was 2 pages long. It contained a single diagram. It changed biology forever. In 1962, Watson, Crick, and a third scientist named Maurice Wilkins shared the Nobel Prize in Physiology or Medicine for the discovery of the structure of DNA. 3 men. There should have been 4. The fourth person, the person whose experimental data was the crucial piece of evidence that allowed Watson and Crick to build their model, whose work was shown to Watson without her knowledge or consent, whose name was absent from the Nobel citation, was a British chemist named Rosalind Franklin. She had died in 1958, 4 years before the Nobel was awarded. The Nobel Prize is not awarded posthumously. This is the most famous story of scientific theft in the 20th century. It is also not quite as simple as it first appears. The full truth of what happened in the laboratories of King's College London and the Cavendish Laboratory in Cambridge between 1950 and 1953 is contested, complicated, and still debated by historians of science. But one thing is not debated. Without Rosalind Franklin, Watson and Crick would not have solved the structure of DNA in 1953, and she received none of the credit. Before we can understand what was discovered and why it mattered, and how it was discovered and by whom, we have to understand what DNA actually is. DNA stands for deoxyribonucleic acid. The name alone is enough to make most people's eyes glaze over, but the thing itself, the physical object, the structure, the reason it matters, is, at its core, something astonishing. Every living thing on Earth, every bacterium, every plant, every insect, every fish, every bird, every human being, contains DNA. It is the molecule that carries the instructions for building and operating a living organism. Not some of the instructions, all of them. Every protein that your body makes, every enzyme that drives your metabolism, every structural element of every cell, all of it is encoded in DNA. The DNA in a single human cell, if stretched out in a straight line, would be roughly 2 meters long. It is coiled and packed in a structure of extraordinary efficiency into a nucleus that is less than 1/100th of a millimeter across. And in every cell in your body, approximately 37 trillion of them, there is an identical copy of the same 2 meters of DNA, copied with remarkable precision every time a cell divides. The information in that 2 meters of DNA is encoded in a chemical alphabet of 4 letters: adenine, thymine, guanine, and cytosine, usually abbreviated A, T, G, and C. The sequence of these letters in a human genome, about 3 billion of them, is the code that specifies you. Not just your eye color or your blood type, your susceptibility to certain diseases, aspects of your personality, your developmental trajectory from a single fertilized cell to a fully formed adult. Understanding this code, reading it, interpreting it, editing it, has become the central project of modern medicine and biology. It underlies every cancer treatment that targets genetic mutations, every gene therapy that corrects inherited disease, every forensic technique that identifies criminals or exonerates the innocent. It all began with the question, What does the molecule look like? By the early 1950s, scientists had known for several years that DNA was the molecule responsible for heredity. The evidence had accumulated through the 1940s, through experiments like the Avery-McLeod-McCarty experiment of 1944, which showed that DNA, not protein, was the material that transmitted genetic information between bacteria. But knowing that DNA carried the genetic code was not the same as knowing how it did it. To understand how a molecule stores and transmits information, you need to know its structure, its three-dimensional shape, the arrangement of its atoms, the geometry of its connections. In the early 1950s, the dominant tool for determining the structure of complex molecules was X-ray crystallography. The technique worked by directing a beam of X-rays at a crystallized sample of the molecule. The X-rays scattered off the atoms in the crystal in a pattern that depended on the arrangement of those atoms. The resulting diffraction pattern, a series of spots and rings on a photographic film, could be mathematically analyzed to determine the molecule's structure. The technique was powerful, but extremely demanding. Producing a good crystal of a biological molecule was difficult. Getting a clear diffraction pattern required exceptional experimental skill. Interpreting the pattern mathematically required years of training, and the gap between a diffraction pattern and an actual three-dimensional structure required both mathematical rigor and intuitive model building. In 1951, two laboratories were primarily working on the structure of DNA using X-ray crystallography. At the Cavendish Laboratory in Cambridge, Francis Crick, a 35-year-old British physicist turned biologist who had not yet finished his PhD, and James Watson, a 23-year-old American biologist who had just arrived from the United States. At King's College London, Maurice Wilkins, a New Zealand-born physicist, and, joining him in late 1951, a young British chemist named Rosalind Franklin. The two laboratories were supposed to be collaborating. In practice, they were competing, and the competition was not conducted on equal terms. The Cavendish Laboratory in Cambridge was, in the early 1950s, probably the most intellectually productive scientific environment in the world. Lawrence Bragg, the Nobel laureate who had co-developed X-ray crystallography with his father, was its director. The culture was one of theorizing, model building, argument, and brilliance. Watson and Crick fit this culture perfectly. Crick was voluble, theoretical, confident to the point of arrogance, and possessed of an extraordinary ability to synthesize information from different fields and see its implications. Watson was younger, ambitious, socially uncertain, and almost shockingly focused on being the person who solved the most important problem in biology. They were told initially to leave DNA to King's College. The Cavendish director had made an arrangement. King's was working on DNA. Cambridge was supposed to focus elsewhere. Watson and Crick ignored this instruction almost from the start, but they had to be careful. They could not simply compete openly with King's. At King's College, the situation was more complicated. Rosalind Franklin had arrived from Paris in January 1951, hired specifically to work on DNA using X-ray crystallography. She was meticulous, technically gifted, and deeply serious about her work. She had spent several years in Paris developing expertise in X-ray diffraction that was among the best in the world. She arrived to find a situation she had not been fully prepared for. Maurice Wilkins, who had been working on DNA at King's for the previous year, had assumed, based on a misunderstanding of her appointment, that Franklin was to be his assistant. Franklin understood, correctly, as her appointment letter confirmed, that she was an independent researcher. The misunderstanding was never resolved. The relationship between Franklin and Wilkins became one of the most dysfunctional collaborations in the history of science. They barely spoke. They avoided each other when possible. The work at King's suffered. Franklin, working with her graduate student Raymond Gosling, made steady and remarkable progress despite this. She was producing X-ray diffraction photographs of DNA of a quality that no one in the world had achieved before. She had identified that DNA existed in two different forms, one hydrated, one dry, and she had begun to accumulate data that would allow her to determine the molecule's structure from first principles. She was careful. She was methodical. She did not want to publish until she was certain. In May 1952, Raymond Gosling took an X-ray diffraction photograph of DNA that would become the most famous scientific photograph in history. He labeled it Photograph 51. The photograph showed the hydrated form of DNA, what Franklin had called the B form, in a clear X-ray diffraction pattern. To an expert eye, the pattern was immediately informative. The distinctive cross-shaped pattern of reflections in the center of the image was a signature of a helical structure. The spacing of the reflections gave direct information about the dimensions of the helix, its diameter, the distance per turn, the number of bases per turn. Franklin, looking at the photograph, understood what it showed. She incorporated it into her analysis. She was building toward a complete, rigorously determined structure, the kind of result she could publish with confidence. She did not publish immediately. In January 1953, Maurice Wilkins showed Photograph 51 to James Watson. He did not ask Franklin's permission. He did not tell her he was showing it to Watson. She did not know it had been shown. Watson later described his reaction to seeing the photograph. He recognized immediately what it meant: the helical structure, the key dimensions. He memorized the measurements before leaving the room. He returned to Cambridge and told Crick what he had seen. Around the same period, Watson and Crick received another piece of Franklin's data, a report she had prepared for the Medical Research Council, which was shared within the scientific community. The report contained precise measurements from her crystallographic work, including the unit cell dimensions of DNA, the key numbers needed to determine the structure. Franklin had prepared this report for internal use. She did not know it was being shared outside King's College. Watson and Crick now had the data they needed. The method Watson and Crick used to determine the structure of DNA was not X-ray crystallography. It was model building. They built physical models, metal plates and rods representing atoms and bonds assembled in 3 dimensions until the structure fit all the available data. It was an approach that Linus Pauling, the American chemist, had used brilliantly to determine the structure of proteins. It required not just mathematical skill, but spatial intuition, the ability to see what a complex 3-dimensional structure might look like before you had proven it. Watson and Crick had attempted to build a model of DNA in 1951, shortly after Watson arrived in Cambridge. The model had been wrong, a 3-stranded helix with the sugar phosphate backbone in the center. Franklin had pointed out, politely but firmly, at a seminar in King's College, that the model contradicted the X-ray data. She was right. The model was abandoned. Now, in February 1953, armed with Franklin's data, they tried again. The result, reached over a period of weeks, was the double helix, 2 strands of DNA wound around each other in a spiral, with the sugar phosphate backbones on the outside and the 4 bases, A, T, G, and C, paired in the center, A always pairing with T, and G always pairing with C. The base pairing rule, which Watson realized almost by accident, working with cardboard cutouts of the four bases and noticing that an AT pair had the same shape as a GC pair, was the key to everything. It explained not just the structure of DNA, but how it could be copied. Each strand could serve as a template for building a new complementary strand. The mechanism of genetic inheritance was visible in the structure itself. When Crick and Watson built the model and saw it was consistent with all the available data, including the critical measurements from Franklin's work, they understood what they had.
Crick told the Eagle Pub.
Watson wrote his sister in America that they had found the secret of life. They also told Franklin and Wilkins, inviting them to Cambridge to see the model. Franklin, characteristically, examined it carefully, asked precise technical questions, and conceded that it was consistent with her data. She was generous in her assessment. She was also, presumably, aware of what she was looking at. The result that she had been working toward, reached by other people using her data without her knowledge. She said nothing about this, at least not in any record that survives. The Nature paper that Watson and Crick published on April 25, 1953, is one of the most elegant scientific papers ever written. 2 pages, 1 diagram. A model of the double helix drawn by Odile Crick, Francis's wife, who was a professional artist. The paper contained one sentence that has become perhaps the most celebrated understatement in the history of science. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. In the same issue of Nature, 2 supporting papers appeared, one by Franklin and Gosling and one by Wilkins and his colleagues. These papers provided the experimental X-ray data that supported the Watson-Crick model. In the order they appear in the journal, Watson and Crick first, Franklin and Gosling second, Wilkins et al. Third. The arrangement implied that Watson and Crick's theoretical model had come first, and the experimental data had been presented to support it. The actual sequence of events, that the experimental data had informed the model, was not stated. Watson, in his 1968 book, The Double Helix, described the events in a way that was revealing and, to many readers, disturbing. He was candid about his and Crick's use of Franklin's data. He was less candid about the ethical implications of this. He described Franklin in ways that were dismissive of her scientific ability, patronizing about her appearance, and condescending about her reluctance to interpret her data more speculatively. His characterization of Franklin was widely criticized and has been increasingly criticized in the decades since. What the book inadvertently made clear, to anyone reading carefully, was that Franklin's data had been decisive. Rosalind Franklin was born in 1920 in London, the daughter of a prominent Jewish banking family with a long tradition of public service. Her father, Ellis Franklin, was a merchant banker who also taught classes for workers at the Working Men's College. Her aunt was a suffragist. Her great-aunt had helped establish trade unions for women workers. She grew up in an intellectual household that valued education highly, though, in the manner of the era, the education of sons was considered more important than the education of daughters. Franklin's father initially resisted her ambition to become a scientist. She became one anyway. She studied chemistry at Newnham College, Cambridge. She completed her PhD on the physical chemistry of coal. She spent 5 years at the Laboratoire Central des Services Chimiques de l'État in Paris, where she developed her expertise in X-ray crystallography and became, by universal account, an exceptionally skilled experimental scientist. Her Paris colleagues remembered her as warm, funny, direct, and enormously competent. When she arrived at King's College in 1951, she was 30 years old. She had a first-class scientific training, international research experience, and a level of technical skill in X-ray diffraction that was exceptional. What she found at King's was a laboratory culture that was, by the standards even of its era, inhospitable to women. The senior common room, where scientists gathered informally, where ideas were shared over lunch, where the social glue of scientific collaboration was laid, did not admit women. Franklin ate lunch elsewhere. Wilkins's failure to understand her appointment correctly, his assumption that she was his assistant rather than an independent researcher, was not simply a personal misunderstanding. It reflected the ambient assumption of the era that a woman in a scientific laboratory was a supporting player. Exceptionally talented, perhaps. A supporting player, nonetheless. Franklin continued her work. By late 1952, she was closer, arguably than anyone else, to determining the structure of DNA. Her notebooks, examined after her death, show that she had independently arrived at key elements of what would become the correct structure. She was planning to publish. She was not yet ready. She did not know that the timeline had been decided for her by someone else's access to her data. After the publication of the double helix paper, Franklin left King's College and moved to Birkbeck College, also in London, where she joined the laboratory of J.D. Bernal, one of the most eminent crystallographers in the world, and a man who recognized her abilities without ambiguity. At Birkbeck, she spent the remaining years of her life doing outstanding work on the structure of viruses, work that was, by all accounts, among the finest structural biology of the decade. She solved the structure of tobacco mosaic virus. She was working on the structure of polio virus when she fell ill. She was diagnosed with ovarian cancer in 1956. She continued working through her illness with a determination that her colleagues found extraordinary. She submitted her last scientific paper from her hospital bed. She died on the 16th of April, 1958. She was 37 years old. The Nobel Prize in Physiology or Medicine was awarded in 1962 to Watson, Crick, and Wilkins, 4 years after Franklin's death. The Nobel Prize cannot be awarded posthumously. It also, at that time, could not be shared among more than 3 people. Whether Franklin would have been included had she lived is a question that cannot be definitively answered, and different people who have examined the question reach different conclusions.
What can be said is this:
The Nobel citation makes no mention of Franklin's contribution. Watson's acceptance speech makes no mention of her. Wilkins's acceptance speech mentions her briefly, describing her X-ray data as complementary to the model-building work. The word complementary does not begin to describe what she contributed. The story of Rosalind Franklin is sometimes told as a simple story. Her data was stolen, she was robbed of the Nobel Prize, she has been vindicated by history. Watson and Crick are the villains. Franklin is the hero. This version is emotionally satisfying and partially accurate. It is also incomplete. The actual history is more complicated. Wilkins's sharing of photograph 51 was not sanctioned theft in the legal sense. The photograph was in the laboratory's files. There were no rules explicitly governing what one colleague could show another. The norms of scientific data sharing were less clearly defined than they are today, and the distinction between Franklin's data and the laboratory's data was blurred by the institutional culture of King's. Watson and Crick did not intend to harm Franklin. They were competing, as scientists compete, aggressively, opportunistically, with their eyes on the prize and less attention to the ethics than the situation required. The rules they broke were moral rather than legal. Franklin herself was a complicated figure. Some accounts from people who knew her describe a woman who was resistant to the kind of speculative model building that Watson and Crick were doing. Resistant to the point of being slow to see what her data implied. Others describe a woman who was systematically marginalized in ways that made it harder for her to do exactly what Watson and Crick did: step back from the data and ask what three-dimensional structure would explain it. The question of what Franklin would have discovered and when, had she been working in a different environment, with a colleague rather than an antagonist, with access to the informal networks where ideas were shared, with the freedom to speculate without the double standard that required her to be more rigorous than her male colleagues because she had more to prove. That question cannot be answered. What is clear is that the environment she worked in, the systematic exclusion from informal spaces, the assumption about her role, the failure of collaboration at King's, were not accidents. They were the product of a scientific culture that did not welcome women as full participants. And that culture shaped what happened.
The data existed.
The question was, who would get to use it? In the 70 years since Watson and Crick published their paper, DNA has become perhaps the most consequential molecule in the history of science, not just as a subject of study, but as a tool. In 1953, DNA was a structure. By the 1960s and 1970s, scientists had begun to understand the genetic code, the precise mapping between sequences of DNA bases and the amino acids they specified. By the 1970s and 1980s, techniques had been developed for cutting DNA at specific points, for copying it, for inserting it into new organisms. These were the techniques of genetic engineering.
In 1990,
The Human Genome Project began, a 13-year international effort to sequence all 3 billion base pairs of the human genome. It was completed in 2003. The genome sequence has since been used to identify the genetic basis of thousands of diseases, to develop new diagnostic tools, and to enable treatments that would have been impossible before. CRISPR, the gene editing technology that emerged from work on bacterial immune systems in the 2000s, made it possible to edit specific sequences of DNA in living cells with a precision and ease that had been inconceivable a generation earlier. Jennifer Doudna and Emmanuelle Charpentier received the Nobel Prize in Chemistry in 2020 for its development. It is now being used to treat sickle cell disease, to develop new cancer therapies, to create disease-resistant crops, and to explore the possibility of correcting inherited genetic disorders. None of this would exist without the double helix. None of the double helix, as it was discovered, and when it was discovered, would have existed without Rosalind Franklin's photograph 51. There is something worth pausing on here. The molecule that has transformed medicine, that is being used to cure genetic diseases that were once death sentences, that will shape the future of human health for centuries, was decoded partly through data that was used without the knowledge of the woman who produced it.
the history of that discovery
Sits beneath the history of everything that followed. Rosalind Franklin is the most famous name that history almost missed. But she is not the only one. Oswald Avery conducted the experiment in 1944 that first proved DNA was the genetic material. He did not receive the Nobel Prize. He died in 1955. 3 years after Watson and Crick's discovery, 2 years before the Nobel Committee might have recognized him. His experiment is considered one of the most important in the history of biology. He is not famous. Erwin Chargaff was an Austrian biochemist who discovered, in the late 1940s, that in any DNA sample, the amount of adenine always equaled the amount of thymine, and the amount of guanine always equaled the amount of cytosine. These ratios, Chargaff's rules, were the direct experimental basis for Watson's realization that A pairs with T and G pairs with C. The base pairing rule that made the double helix comprehensible was not Watson's discovery. It was Chargaff's. Watson met Chargaff at a dinner in 1952. Chargaff was not particularly impressed with Watson. Watson did not fully understand the significance of Chargaff's rules until later. Raymond Gosling, who actually took photograph 51, received credit in the Franklin and Gosling paper, but is rarely mentioned in popular accounts of the discovery. Nora Barnard, Linus Pauling's secretary, typed the letter that, had Pauling received it and understood its implications, might have allowed Pauling to solve the structure before Watson and Crick. The letter was intercepted by American authorities who suspected Pauling, a peace activist, of being a communist sympathizer and had revoked his passport. Without the ability to travel to London, Pauling could not see the King's College data. The structure of DNA, in other words, was decided partly by McCarthyism. History, when it tells the story of great discoveries, tends to compress and simplify. It finds heroes and gives them names. It finds moments and calls them turning points. It is easier to remember Watson and Crick than to hold in mind the dozens of people whose work made theirs possible. But the compression is always partial. The simplification is always a choice about whose story to keep and whose to let go. Let me try to say what DNA actually means.
Not the chemistry,
Not the medicine. What it means as a fact about the world. Every living thing on Earth shares the same genetic code, the same four-letter alphabet, the same system for translating sequences of bases into proteins. A bacterium and a blue whale are using the same coding system, a system that was established in the common ancestor of all life on Earth, roughly 3 to 4 billion years ago. This is what the structure of DNA revealed once biologists understood it fully, that life on Earth is not a collection of separate experiments. It is one experiment, running continuously for 4 billion years, copying itself with imperfect fidelity, imperfect enough that mutations accumulate and evolution happens, faithful enough that the core machinery is preserved across every living thing that has ever existed. You share approximately 60% of your DNA with a banana plant. You share 99% with a chimpanzee. You share 100% with every other human being in terms of the basic structure of the genome, though the specific sequence varies enough, about 0.1% of the 3 billion base pairs, to make every person genetically unique. The same molecule, the same alphabet, the same code. Life on Earth is a family, and DNA is the language the family shares. Rosalind Franklin spent her career trying to understand the structures of molecules, not for abstract reasons, but because she believed, with the particular conviction of a great experimental scientist, that structure was the key to function. That if you could see how something was shaped, you could understand what it did and how it did it. She was right about DNA. She was right about viruses. She was right about the principle. She did not receive the prize. She received something more durable than a prize. She received a place in the permanent record of how the world came to understand itself. Not the place she deserved, not the acknowledgement she was owed during her lifetime, but the place history eventually assigns to people who did the work honestly, carefully, and without compromise, even when the world they were working in made honesty and care harder than they should have been. The double helix stands, the photograph exists, the data was real, and the molecule, indifferent to the arguments of the people who study it, keeps doing what it has always done. It copies itself. It passes the information on. It remembers. On a clear day in Cambridge, if you walk down Free School Lane past the Cavendish Laboratory and then along to the Eagle Pub on Bennett Street, you can order a drink in the room where Francis Crick made his announcement. There is a plaque. It commemorates the discovery of DNA. It mentions Watson and Crick. Across the city, at King's College London, which is in London, not Cambridge, there is a building named after Rosalind Franklin, opened in 2000. There is a lecture theatre named after her. There is a Mars rover named after her, launched by the European Space Agency in 2022, exploring the surface of another planet for signs of life. The signs of life it is looking for would, if found, almost certainly involve something like DNA. Franklin did not live to know that her photograph had been decisive. She did not live to know that the discovery of the double helix, her double helix, arrived at with her data, would transform medicine, forensics, agriculture, and the understanding of life itself within decades of her death. She did not live to know that she would eventually be recognized, that the textbooks would change, that the historians would do their work. She just did hers. In her laboratory at Birkbeck, working on the structure of tobacco mosaic virus, surrounded by colleagues who valued her, in the final years before her illness made working difficult, Rosalind Franklin was doing what she had always done. Looking at the patterns the light makes when it passes through matter, trying to understand what they meant, trying to see the shape of things. She was very good at it.